Hello, everyone. We are welcoming all of our attendees for today's webinar. All right. Okay. So for today, we are actually super excited to have our speakers. So we have a full program today, and we have two speakers from Nice in France. But before that, let me just start with our agenda for today. So first, I will introduce muscle bias systems, intro about microelectrode arrays, which we provide here at Muscle Bias Systems, and then we will proceed to the main program by Professor Dr. Michele Studer and Dr. Michele Bertacchi with their talk over here. After that, we will go to our Q&A session. So of course, you can add your questions already under the Q&A section anytime, but we will only go through them after all of the talks. So we will not go through the questions during the talks themselves. So please feel free to already add there all of your questions. If in case you have them, we will go through them one by one afterwards. And then if in case you would like to just say hello, nice to see you, just use the webinar chat that is right here. Also, if we have some things like, papers to download or some files to download, please feel free to take a look at the webinar chat, because this is where we add all of these links that you can just click on, and then you can download, for example, protocols or the paper that will be presented by both Michelle and Michele. All right. Okay. So nice to see you, everyone. Thank you again for joining us. Now I'll proceed to the introduction. So welcome to our Maxwell webinar. My name is Maria Bien. I will be your host for today, and I'm happy to show you how to unlock the full potential of multi-well high-density microelectrode arrays. So first, about muscle bias systems. So we are a global leader providing next-generation electrophysiology platforms. So it's really our mission to empower all of our users, from academic laboratories to pharmaceutical companies, on their functional assays, especially like today organoids, but also 2D cultures and other electrogenic samples. And our company is based in Zurich, Switzerland, and we cater to hundreds of laboratories around the world. And we are very proud that our systems have been used in already more than 200 publications by our users, many in top journals, such as follows. So it is really our honor to be part of this work. And aside from really developing and innovating new products for electrophysiology, we're also very happy to build this network of researchers and scientists working on electrophysiology together. So thank you so much. Now you're also part, and welcome to this webinar. So what are these neuroscience applications where our systems can be used for? As I mentioned, 2D cultures such as iPSC-derived neurons and primary neurons, 3D such as organoids and spheroids, also acute samples such as retina and brain slices. And for today, we will focus more on organoids, and that is what the talk will be focusing on by Michele and Michelle. So now about muscle bias systems technology. Here, our motto is every cell has a story to tell. And to characterize neurons, the popular approach is basically imaging, to see your cells, to look at the different cell types, to see how they're connecting, how they're growing. And also on the other side, the gold standard for electrophysiology is patch clamp. Now, our technology is something in between. And these are high-density microelectrode arrays that allow us to also capture this electrical activity of the neurons, such as here below. But aside from that, also to see how this signal propagates across all of its axonal arbors, such as what you can see here, an action potential initiating and propagating along the axons. In that sense, you are actually able to do electrical imaging using this technology. So how is that made possible? So this is our core technology. So as you can see here, this is how the microelectrode array looks like. This high-density microelectrode array is a 2 by 4 millimeter square, consisting of, in total, 26,400 electrodes. These are really very tightly spaced together, that in a millimeter square, there are roughly around 3,300 electrodes. Each of these electrodes are highly sensitive readouts. And aside from that, we can also use any of these electrodes for flexible electrical stimulation. Now, looking at this more zooming in, you can see that the distance between electrode to electrode, center to center is 17.5 micrometers, which allows us to actually capture the signal of every cell across multiple electrodes, such as this one, which is how we are able to do this accent tracking assay. All right, so more about how it looks like in terms of the plates. So we have multiple versions, the single well max 1 chip with 26,400 electrodes, the max to 6 well plate, and the 24 well plate, once again, each of which has the very same 26,400 electrode high density microelectrode arrays. It just multiplies to 6 and then 24. And how is it in terms of systems? So the Max One system is very versatile that you can use inside of your incubator or on top of your desk for with perfusion system. You can also combine it with other equipment. And here we have the Max One chips compatible, while at the same time for higher throughput experiments, we have the Max Two, which can cater to 6 and 24 well plates. And it has a built-in incubator with a heater at the bottom and on top, so this closes. And there's also a CO2 input that maintains a good environment for your cells during recording. So aside from that, of course, it's important to interact with the device using the software. And we have MaxLab Live Suite, which is our all-in-one software solution that allows us to visualize the data, record using the assays, analyze these recordings, and also export these metrics and reports in a very easy manner. And just to elaborate more on how to record using assays, we have developed these 4 different assays focused on neurons, activity scan, network, accent tracking, and stimulation. If you would like to learn more information about this, just contact us. But I think both Mikel and Michelle will show more on these, on how these are actually used in their work. Okay, so just to elaborate more on neural models, how 3D neural models, how our system has been used for that. So of course, there are different methods for recording organoids. You can do whole organoid in culture or acute. You can do sliced organoid in culture. You can also use air-liquid interface in culture or acute. And these are just a selection of some of the works already done with organoids using our system. And if you want to know more, just go to this link right here. And for today, thanks to Michelle and Michele, we are highlighting this work in eLife. Was just published this month or last month, actually. So we are really looking forward to see how they used Max One for this specific work. Looking forward to that soon. Okay, so at Maxwell Biosystems, we always make sure that the solutions that we provide our users really make sure that this can be used for maintaining good health of the organoids and also allowing for many different types of experiments. So we want to make sure that the condition of organoid is really good during the study, that the culture media that we recommend is good, that there is acclimation of the organoid to the recording environment, which is on top of the microelectrode array, better if it's inside the incubator while you're doing the experiments, and also the age of organoid is important. Additionally, we want to make sure that the quality of data to derive conclusions is really high, meaning that we can actually identify that the information that we are getting is real biological information and not just noise, that we have access to single cell information, but also subcellular if in case it's useful. And also this is made possible by our sensitive readouts. And finally, to be able to also allow different types of experiments, including longitudinal studies, like development of activity across multiple days, and also allowing pharmacological application if in case this is needed by the study. So overall, to support this, we have developed brain organoid plating guidelines, protocols, and also supplementary guide in order to help you in making sure that your organoid experiments are working very well. And we also have new instruction videos that looks like this, just showing you how to place the organoid, how to attach it, how to maintain it, and how to make sure that it's working very well with our high density microelectrode array. So if you would like to learn more, I think the link can be shared here in our chat to get access to this. All right. So now let me proceed to some results using our system thanks to our multiple users. So we have many different types of organoids already recorded, both using activity scan and network assay. And yeah, so that's it from muscle biosystems side. Now I would like to introduce our two speakers of today. I think you can see them already on your screens. So yes, so first of all, let me introduce Professor Dr. Michelle Studer. So Michelle is a well-established scientist in neurobiology with extensive expertise in mouse developmental biology and more recently in human brain development. She received her training in leading laboratories in London, UK, including a postdoctoral fellow at the National Institute for Medical Research as a junior group leader at King's College London. At King's, she was awarded an MRC Career Development Award to establish her own lab. And in 2000, she broadened her expertise as a visiting scientist at UCSF in the US. So subsequently, she was recruited as a principal investigator at the Telethon Institute of Molecular Medicine, or TIGEM, in Italy. And then in 2009, she moved to France as an interim research director and group leader, receiving an ANR Chair d'Excellence Award to investigate the molecular and cellular mechanisms underlying brain organization in physiological and pathological conditions. So that is Michelle. And I would also like to introduce Michele, because they will do the presentation one after the other. Dr. Michele Bertacchi is a researcher in developmental neurobiology at INSERM and a member of Dr. Studer's lab at the Institut of Biologie Valrose in Nice, France. So he earned his PhD at Scuola Normale Superiore in Pisa, Italy, under the mentorship of Dr. Federico Cremisi, focusing on the role of morphogens in directing retinal versus cortical fate in mouse embryonic stem cells. So now we will focus on the work of both Michele and Michelle in the next presentation, which is entitled Modeling Normal and Pathological Brain Development and Function Using Human Organoid Systems. So please welcome first Dr. Michelle Studer. Hello, Michelle.
Hi, everybody. So, first of all, thank you very much, Marie, for this nice introduction. And so the title you know already. So the plan is that we do a kind of general introduction about two projects that we are following in the lab. Using organoids and using the MEA system. And Michele, who is doing the real job, and actually not me, is going to more talk about in more details about how we use the MEA in our organoid system and the beautiful results that we got. So here we are. Doesn't work. Okay, fine. First of all, I just wanted to introduce our institute very quickly. So we are located here in Nice, which is south of France. Paris is here on the French Riviera, so it's a really nice place. And we are a research and affiliated institute supported by our university called Côte d'Azur, CNRS and INSERM, which are kind of two governmental research institutes, a bit equivalent like the NIH in the States. We are 300 members, 25 nationality, 26 teams, 3 startups, and 11 technical platforms. So our campus is actually a historical site. This is where here is the president, where there are administration staff, so not our labs. And this is actually the more modern building where our labs are located. We are interested generally, we're a multidisciplinary institute. We're interested in basic research and in development biology, cell biology, neurobiology, genetics, and so on, but also focusing on translational research and are really studying several kinds of pathologies like metabolic diseases, neuronal diseases, cancer, and so on. And the strength of our institute is that we are using different experimental models from sea urchin to C. elegans, Drosophila, zebra mouse, and recently human cells and human organoids. So we are interested actually in kind of connecting, if you want, physiology and pathology, which are two processes that are very tightly linked. Indeed, we need to understand how mechanisms work in order to get more about the physiological mechanism of disease, but pathology can also help us in understanding more about what's happening physiologically. So we think these are two very much linked processes. So today we are going to talk about two projects that we have in the lab. The first, as already mentioned by Marie, is about how FGF8 actually acts in regional identity in human cerebral organoids, and this has been recently published on eLife. And here you see a kind of organoid with glutamatergic and GABAergic neurons that are divided. And in our second kind of project, it's more pathological, and we are interested in understanding how glioblastoma cell lines actually invade a normal physiological cerebral organoids, and in terms of also molecular mechanism, cell behavior, and so on. This is unpublished work, and we are doing this in collaboration with one of our IBV colleagues. So very quickly, for the ones who are not super familiar with cerebral organoids, so cerebral organoids derive from iPS, so induced pluripotent stem cells, which can be reprogrammed by skin or by blood, and they are grown in a kind of bigger or smaller bioreactor, and at different stages. I mean, during organoid development, now the Arlotta lab has even organoids for 7 years or even more. And the beauty of this organoid system, if they are cerebral organoids, the idea is to mimic as much as possible early cortical development, for example, early brain development, at the physiological point of view, meaning just we want to know more about how human brains develops. And we have this kind of in vitro model, but also at the level of disease pathology, of genetics of disease. And obviously, this is a wonderful platform for drug screening. There are many applications of cerebral organoids. The beauty of it, you can see actually it looks like a ball of cells with all a kind of what we call the rosette outside, but also here in green, also neurons, I mean differentiating neurons, is that compared to 2D, they have a wonderful 3D cytoarchitecture. So with kind of all layers, and we can use them for studying brain development, for studying brain diseases, for understanding brain regionalization, and we can do transcriptomics on organoids, drug screening, and what we can see also, see later by Michele, also record spontaneous electric or induced electric activity. So our first project today is about how FGF8 actually acts in regionalization. So very quickly, FGF8 is a very known morphogen, which diffuses from, okay, you can see here, from the anterior part of the brain, and it interacts with other morphogens like BMPs and WNTs or sonic hedgehog in order to pattern the forebrain and also during the growth. Now, however, FGF8 during early development is not only expressed here in the future telencephalon, but also at the level of diencephalon and at the level of the mid-hindbrain boundaries, the isthmus, and this makes it very difficult to understand its unique role, since animal models lacking FGF8 or overexpressing FGF8 gave really a difficult results in terms of phenotypic analysis. Also because FGF8, as I said before, interacts also with other morphogens like BMP, SWINT, and so on. So we decided that we wanted to study the unique role of FGF8, first of all, in human cells, because this was not really done before, but also in organoid and in human regional telencephalic, to understand its role in human regional telencephalic identity. So to do so, I say we, but actually it's Michele, from IPS, so kind of commercialized IPS, that we used a protocol in which we inhibit WNT And thanks to the protocol, the rosettes that you can see here, there are SOX2 positive, but there are also FOXG1 positive. And this was very important for our protocol and for our study, since we wanted to have rosette of telencephalic identity. And this is what we managed to do. And with our protocol, we managed to keep, compared to a control without Wnt inhibition, telencephalic identity. So next step, we added FGF8, and actually we tried to optimize the dosage, to optimize the time when we added. To shorten it, we actually added at day 10 of culture until day 50-60. And we tested obviously the efficacy of FGF modulation, and we found that most of the FGF targets were overexpressed, but importantly also one of the top target of FGF direct target, which is NR2F1, was nicely inhibited in our rosettes, in progenitors, but also in neurons. And importantly, this was maintained during culture. So after we did some single cell RNA sequencing with Wnt inhibition by adding FGF8. And as you can see here, the UMAP look quite different. Here we identified neuronal cells of neuronal identity, and here are cells of progenitor identity. So what we found is that upon FGF8 treatment, we had here a reduction of the number of these cells. These cells are of glutamatergic identity, as seen by these markers like EMX1, UD6, or VGLUT1. And on the contrary, we had an increase of these clusters of cells. We turned to be GABAergic progenitor and neurons, as you can see here with ACL1 before MASH1, DLX1, and GAT2. And if we looked at organoids, we indeed could confirm this data, and we saw an increase of VGLUT2 of glutamatergic identity in control, while a decrease of VGLUT2 in FGF-treated cells, and an increase of GAT67, which is obviously a mark of GABAergic. For the aficionados of development neurobiology, what we found is that actually cortical identity took not only a GABAergic ventral identity, so the dorsal cortical identity, but even more specifically a lateral ganglionic eminence identities with kind of striatal-like fate. So to sum up this part, we found that FGF8 is an inducer of anteroventral telencephalic identity, and that actually in our organoid, by adding FGF8, we had over-representation of GABAergic interneurons, as you can see here. Now, obviously, the next question was if we found this in terms of markers, in terms of cell type, but what happens functionally? And this is the part that Michele will talk about. And the question is, does the imbalance between glutamatergic and GABAergic neuron can affect spontaneous electric activity within the organoids? The second project I would like to talk about and introduce is about glioblastoma. Cell behavior in human cerebral organoids. So glioblastoma multiforme GBM is actually the most aggressive and common brain tumor, arises from glial cells, mainly astrocytes, has an intratumor high heterogeneity, and what is quite bad is that 90% of patients with glioblastoma face recurrence. It's difficult to find a real, at the moment we don't have a real cure. So this work has been done in collaboration with Thierry Virol, our colleague at IBV. And what he showed before is that in collaboration with clinicians, he took some patient-derived glioblastoma cells, added the GFP, did produce some tumor spheres, and actually transplanted this tumor sphere into mice, and saw that actually they were developing high tumors and big tumors and aggressive tumors. In this tumor sphere, obviously molecularly, it's very heterogeneous. And one of the genes that are expressed in this tumor sphere is SELF2. SELF2 is a member of the self-Bruno-like family of RNA binding proteins, is strongly expressed in proliferated glial stem cells, so OLIG2 cells, confers a malignant potential to glioma stem cells, and is associated with high tumor potential. When the group of Thierry inhibits through short hairpin SELF2 in this tumor sphere, what he could find is that actually after transplantation into human brains, he could find that the tumor potential was actually very much decreased. And he concluded that SELF2 downregulation represses actually stemness markers. And this work has been published last year. I wanted to actually, this is our last retreat. So I'm not sure if you can recognize me, but you probably can recognize Michele. Here, he will take over. I just wanted to thank our collaborators, specifically Thierry Viral, our platforms, and our funding agency. And Michele, the stage is yours.
Okay, so I'm gonna start here. Thank you so much again for organizing this. Thank you so much, Michelle, for the introduction. I will just show some reminders about the two projects. So the first one that Michelle showed is about FGF8 signaling. And as Michelle said, our question was, can we understand the specific roles of FGF8 on the regional identity of human neural progenitors and neurons, focusing on the telencephalon. So in the next slides, what you will see will always be a comparison between our control condition, that is the Wnt inhibited organoid, that as you saw, they're acquiring a fate of dorsal telencephalon, so neocortex, and the treated condition is just that we add FGF8 to the culture medium. So we are gonna compare these two, as simple as that. Also, just to remind you that normally our control Wnt inhibition organoid, they originate a lot of glutamatergic neurons. So this is a dorsal telencephalic identity. They also have some GABAergic neurons, but this is a very low percentage, as you can see by GAD67 staining. Vice versa, when we treat the organoids with FGF8, you can see that we have a huge increase of GABAergic neurons. So basically what we found many different things. We don't have time to go into detail, but one of the main actions of FGF8 was to modulate the dorsoventral identity of progenitors and neurons, and basically to ventralize the organoids and induce a ventral GABAergic identity at the expense of a dorsal glutamatergic ones. And as also Michelle said, now the question was, what is going on at the functional level? Is this different ratio of dorsoventral neurons giving rise also to a different activity? And this is where we use Maxwell technology. In the lab, we have the MAX1 system. Marie also said that there are different strategies to put organoids on the multi-electrode array chips. In our case, the beauty of the 3D organoids is that they have a higher cellular heterogeneity and they form very nice network. So what we try to do is to not dissociate the organoids. We are not cutting or slicing them. We try to keep as much intact as possible. But to make them stick on the multi-electrode array surface, we use a triple coating with polyornithine, laminin, and matrigel. So in 1 or 2 weeks of time, the organoids are attaching to the surface, and we can see that neurons are elongating axons on the side. So if we do the same in parallel on a normal plate and we look under the microscope, we will see the organoid, basically, you can see the edge here, and a lot of axonal tracts that are elongating all around. Then when we record the activity of the organoids in the Maxwell system, this area that are all around the edges of the organoids with a lot of axonal tracts are also the area in which we find most of the activity. So you can actually guess the position of the organoids on the chip. Okay, let's go back to the sample. You remember we were comparing Wnt inhibition control organoids. We did this at different stage of development. These are 4-month-old organoids and 7-month-old organoids. We found a nice level of activity, but when we did the same with FGFA treated organoids, we could find that there was a lower level of activity. This was quantified by the Maxwell software. There's a lower percentage of active electrodes, but there's also a lower firing rate and spike amplitude. So this means that upon FGFA treatment, we have a lower activity rate, which is probably compatible with the presence of a lot of GABAergic neurons, as we saw before. So even more interesting when we went at the network level, this is a typical activity of our control organoids and also the typical activity of cortical organoids. What does it mean? You can see that in this raster plot and also in the video, we have some action potential, we have some random activity, but then they tend to fire all together, right? So what you will see in the raster plot is columns, which are called bursts. In each burst, you have all the spikes of the neurons that are active at the same time. This is very important because it's kind of a readout of the maturation of the network. It was shown in vivo in fetal development, but also in vitro with cortical organoids, that when the neural circuits are forming, neurons start to fire all together. So you have this kind of synchronization of the activity and oscillation of the activity. And this is what we also find in our organoids. However, when we did the same with FGFA-treated organoids, the situation was quite different. So you can see that they're still active, but the organization of the activity in time is quite different, right? So we can still detect some burst, but you can see each burst has not many spikes inside of it. And if you look at the space between different bursts, there's a lot of random activity. So this was also quantified by the Maxwell software. So basically you can see that the spikes that are falling inside the burst are getting lower. And if you look inside the burst, you have a lower number of spikes that are constituting the burst, and also the firing rate in terms of frequency is going down. So this means that with FGFA-treated organoids, we have a lower synchronicity and a higher random activity. And this made sense for us because when we went back to the literature, we found that when people are plating glutamatergic neurons, so cortical neurons, they get this nice activity with this beautiful burst that you saw before and very low noise inside between the bursts. However, when you change the ratio of neuronal type and you increase the ratio, you increase the number of GABAergic inhibitory neurons, actually the situation changes a lot. You still get burst, but you have a lower number of spikes inside the burst and a higher number of spikes that are falling outside. So this random, less synchronous, less organized activity. This is exactly the same that we found with our organoids, suggesting that GABAergic activity was responsible for this. Now, to be absolutely sure about this, we decided to take another approach. Can we block transiently the activity of GABA inside our organoids? So this is an example of a FGFA-treated organoid. You can see as before that we have nice spikes, but we also have a lot of non-synchronized activity in between the spikes. So what we decided to do, we're gonna block GABA by using bicuculin, and this in theory should give either more activity or a better synchronization of the activity. And in our case, we found especially a better synchronization of the activity. This is the same organoid, like 15 minutes after treating with bicuculin, and you can see that now the spikes, the bursts, sorry, are getting more strong with a higher firing rate, and also we can see also in this high magnification, we have kind of a cleaner space in between the bursts, meaning that the random activity is going down. Or if you see from the other point of view, we have a higher percentage of spikes which are falling inside the burst. So this means that GABA is responsible, at least in part, for this decrease of network synchronicity in 3D organoids when they're treated with FGF8, because we have a lot of GABA neurons there. Last but not least, we also use the axon propagation assay. Basically, already by looking at control organoids, something that I think is interesting is that you you can see like four-month-old organoid and compare with 7-month-old organoid, and you can see that the structure of these neuronal networks is becoming more complex. You have a higher branching, you have a higher ramification. So basically what you can do with this system, you can follow in time, in a longitudinal way in vitro, the maturation of the neuronal network. But now let's go back again to our FGF8 story. This is what we found. You can see the initial signal, but then you can see that this signal struggles to travel around. So we have a kind of decreased propagation of the signal. Again, we can quantify with all the different kind of parameters that the Maxwell software allow us to access. And we have a lower, a shorter axonal length, branching length, etc. Also, the latency of the signal is really shortened. And also the amplitude of the signal at the initiation site is getting lower. So this means that these GABAergic neurons are kind of responsible for an intrinsic inhibition that is kind of reducing the propagation of the spontaneous signal along the axonal tracts. Okay, so to conclude my first part, we found out that FGF8 has multiple roles. The main one is that of an inducer of ventral GABAergic identity in human neural progenitors and neurons. So as a result of this, we have a different ratio of dorsal glutamatergic versus ventral GABAergic neurons, with a lot of GABAergic neurons when we treat with FGF8. And with the multi-electrode array technology, we could also see what is the consequence of this on the activity. We found a lower activity rate, a lower synchronicity, and a decreased signal propagation along the axonal tracts. So I think this was the most interesting part for us, because we could go, thanks to the multi-electrode array, at a functional level. So here we can correlate on one side the regional identity of the cells, or the composition in terms of dorsoventral cells in the network, and on the other side, really the real consequence on the spontaneous network activity in 3D human organoids. Well, as Marie said, we now recently published this paper. So for further details, I invite you to go and give a look. And I will use the last minutes of the talk to switch to the second project. So with this, I concluded. This is our team. You already saw us before in the picture. And I want to say a big thank not only to the whole Maxwell community. I'm really learning a lot. We are not electrophysiologists, so it's really nice. We are really improving a lot. And for the analysis of what we cannot understand and go into depth, we are really enjoying this new collaboration with TJ. So thank you so much. And I think that now we are going to switch to questions.
Thank you so much, Michelle and Michele. Really, really great to see your different models, starting from F8 and now with the glioblastoma model, and even more in the future. Really, really looking forward. Thank you so much for sharing this work. So now let's go to the questions. Okay, so let's start with Claudia. Thank you for your question, Claudia. So what age of cerebral organoids do you recommend for recording? Is there an optimal age?
Right, I think, Michele, you can answer because we tried different ages.
Yes, yes.
Yeah, so it's a wonderful question. I think Claudia is really right to ask this, because the age is really important. Of course, there's a lot of variability because it depends on the protocol. In our hands, so I'm going to answer for our protocol, we tried starting from 3-month-old organoids. And there was, at 3-month-old, we started to have some activity. 4 months, it's going to be better. And so in the data that I showed for the FGF8 project, we had 2 main temporal windows. So we were focusing on 4-month-old organoids and 7-month-old organoids. But this doesn't, I don't want to say that it's not possible to do that before, because it depends a lot on the protocol. So you need to check and to optimize depending on your protocol. Yeah.
Thank you so much. So she followed up that there are 100 or 200-day-old organoids in the presentation. So I think you already covered that maybe with other protocols, also recording younger ones could work as well. Thank you. So JP, in the MEA results, You see most of the activity in the axons elongated from the organoids. Do you think this is representative of the whole organoids network activity? Thank you.
We always get this question. Well, the recording is by definition 2D, right? Because you're recording from the
That's true.
surface of the chip. And in our case, it's also really all around the organoid. So these axonal tracts that are elongating around, we don't know why. Now we are trying different plating strategies to also get signal from the inside of the organoid. I think anyway that it could be representative anyway of the whole organoid network because they're connected. And so even if you're recording only from this surface and from the outside part, the signal is also coming. I mean, the réseau, the network of the neuronal network is also coming from above, from the part that you cannot access during the recording. And by the way, when we look at the activity of these organoids, sometimes we see really waves of activity. It starts from one side, it goes to the other side. So even different organoids can be connected. When I showed, maybe I forgot to mention that we put more than one organoid on the surface. So it's always like 2, 3, even 4 organoids, and they tend to connect together. So they told me that this is better defined as a connectoid. So yeah, I think that you, even if you record only from a subset of the axonal tracts, you can something that is really representative of the whole population. This is my feeling.
Thank you so much, Michele. So now we have more and more questions coming up. So from Wissam, thank you for the question. Do your cerebral organoids contain microglia? Both Michelle and Michele, feel free to answer.
Beautiful question. Yeah, so as I mentioned at the end of the presentation, we are really trying to scale up the complexity of the system. So you want to have something that is in vitro, is simplified, easy to access, but also at the same time, you can try to scale up a little bit the complexity of the system. We are adding endothelial cells. We also have astrocytes, but not a lot, and we have to wait for a longer maturation time of the organoids. We don't have microglia. So there are a lot of protocols to produce organoids with microglia. Very often you produce microglia separately and then you add microglia cells on the organoid. So for the moment we don't have it, but here it's something that we have in our mind because especially for the glioblastoma project, here we're dealing with cancer, and so to have some kind of immunitary system inside the organoid would be very interesting, of course. Yeah.
If I can add something, I mean, you said we don't have microglia, but we haven't really checked with markers of microglia. So maybe actually we should at later stages.
I would be surprised, but it's
I would be
sure that
surprised as well, but I think we should, yeah. So for the moment, we haven't really looked, but most probably not.
Thank you so much. From Francesco, I think he got excited by the collaboration also with TJ. Thank you, by the way, for mentioning about our Maxwell Summit. So what do you use for spike sorting?
okay, so
Do you know, or should we ask TJ?
I'm not the right person to answer to this. I'm sorry. So what Francesco can do is to contact TJ. He is really the expert. By the way, I think it was TJ publication on Nature Communication featuring Maxwell system and also on Maxwell material that you can find on Maxwell internet site. There's also some data about connections in different parts of the organoids. If I will remember, it was done in that paper, but I could be wrong. But if I will remember, it was that paper. So you can look at what he used because it was a new software that he created, and you can for sure contact him or you write to us and I will ask and I will give you more details. I'm so sorry, I don't have the answer ready for you.
Thank you so much. Yes, once again from Wissam, excellent talk. So have you looked at the state of the GABAergic neurons in the GBSH control organoids? So she's wondering if the epileptic activity we showed is due to the death of the GABA cells or some sort of selective inhibition.
But if I can answer.
Yes, please go ahead.
I think this is an excellent question, actually. We haven't looked if any GABAergic, if the death of GABAergic. To be honest, we don't think so because activity comes back. As soon as after we edit the cells, we have a shutdown of activity. The system kind of try to re-equilibrate, and then the activity comes back. So it's worth checking, but I would expect not.
But it's true that we never did a staining for GABAergic markers in the organoids when we detach them,
Yeah.
because it's true that if in the control organoids we have a small percentage of GABAergic neurons, it's a small percentage, but they're important to refine the network activity. And it's true that if the glioblastoma is attacking specifically these cells, it could be responsible for this kind of remodulation of the activity and to induce this epileptic activity. It could be, I mean, epilepsy is also caused by an imbalance between glutamatergic and GABAergic
Yeah,
activity. So
imbalance, yes.
it could be an idea for a mechanism. I think this is worth to be investigated. So thanks so much for the question and the suggestion. It's really interesting.
Wonderful. Okay, so thank you so much. Claudia said thank you so much as well. So I think that's it. Are there any other questions from our audience? Well, if not, thank you so much. So maybe I can just share a little bit about our next events. So we would like to already announce to everyone that we have our next webinar on January 22nd. So we will have Mitsubishi Tanabe Pharma in the US with two speakers as well, from Dr. Makoto Tamura and Dr. Aboukadir Toychev, and with Laura as our host. So very much looking forward to that. And if in case you got our postcards, thank you so much for sharing with us different pictures from around the world. If you have your own, please share it with us as well. And these are the two events that we have this month. One in Cold Spring Harbor in the US and one in ISSCR in Singapore. So see you there if you're in the area. And finally, these are all of the other events where we are at. So thank you so much, everyone, for joining us today. Really happy to have you all. Thank you so much, Michelle and Michele. Very, very nice to hear your talk today, and looking forward to talking more with you in the future. Thank you so much. And
Thank you very much, Marie,
yes,
about hosting us and giving us a possibility to show you all our data. So thank you to all of the audience and to the great question. Thank you and have a
thank
great
you. Thank
evening or a great day.
yes, yes. Have a nice December and happy holidays soon. Yes. Thank
Thank
you.
you.
Bye bye.
Bye.
Bye bye.